Hello guys, gals. Let's get started today with our discussion on glands and membranes. Our last discussion that when we were in class was on epithelial tissue, so we talked about how stratified and simple means multiple layers in one layer, and then we talked about shapes of the cells, and then we're to find those epithelial tissues in the body. And one of the things we emphasized is that most of our glands are made of epithelial tissues. And as we look at these epithelial tissues, since they form membranes, we really need to talk about the junctions or the connections between the cells to talk about how the membrane can function. So when we think of a cell junction, I want you to think of something that connects two cells to each other. So as we're looking at this cell junction, the cells need to be anchored into the extracellular matrix so that we're going to have proteins embedded within the cell that anchor to the extracellular matrix. And then we use these cell junctions also to communicate with other cells. So one cell will use a cell junction to communicate with another cell. And probably the most commonly talked about cell junction is the tight junction. As its name implies, it is, well, really tight. And these tight junctions are small zippers. You can think of them as a waterproof zipper between our cells. And these waterproof zippers are going to seal off the intercellular space. It makes it very difficult, exceedingly difficult for liquids and substances to go in between the cells. When we use tight junctions, generally speaking, we want a waterproof membrane. So when you think tight junctions, I want you to write this down in the margin notes right now on your PowerPoint slides. Tight junctions equal waterproof membranes because they don't let substances, liquids, go in between the cells. We also have desmosomes. And in desmosome, unlike a tight junction, which focuses on waterproof membranes, desmosomes focus on resisting mechanical stress. So when we have lots of stretchy membranes or membranes that need to resist lots of torsion and torque, there's going to be a lot of desmosomes present in that membrane. Now when we look at a desmosome, we're going to zoom in right here. And for some weird reason, I am not sure why my taskbar is not going away on the bottom of my screen right now. Hold on, let's click on PowerPoint. Let's go to slideshow. Yep, I don't know what's going on. All right, anyways, continuing with the recording. So as we look at that desmosome right here, we can have some anchoring proteins. These anchoring proteins are actually going to connect to the cytoskeleton of the cell. And then over here, we have more anchoring proteins that have connected to the cytoskeleton of the cell. Let's see here. Pointer. I really want... Here we go. Oh, yeah, no, this isn't working well at all right now. I digress. And as we look at these hemidesmosomes, they are going to, or these desmosomes, they anchor cells together. I really want to emphasize that to you. We also have hemidesmosomes. I don't have a picture of a hemidesmosome on the slide, so I'm going to skip ahead a couple slides for you. And on the bottom of the screen right here, we have a hemidesmosome. And this hemidesmosome is going to be kind of like half of a desmosome. Hemidesmosomes typically are going to link an epithelial cell to the basement membrane underneath. And then finally, we have gap junctions. Gap junctions allow for liquids, cytoplasm, for chemicals to go from one cell to the next cell. So when we think of a gap junction, I want you to think of a communicating junction. These let chemicals, substances go in between cells. And I really need to emphasize this for you right now. Our cardiac muscle tissues have lots of gap junctions at the intercalated disc of the cardiomyocytes. We'll talk more about that when we get to the muscular system. When we look at our lens and our cornea, the lens of our eye and the cornea of our eye, these have living cells in them. But we can't have blood vessels in our lens and our cornea of our eye because if we had blood vessels, that would block light from getting into our eyes. So instead what we have is a whole bunch of gap junctions in the cells of the lens and the cells of the cornea of our eye that allow for nutrients to be efficiently moved through the lens and cornea cells to keep our lens and cornea from dying. So here's a figure from your textbook. It has the desmosome, the hemidesmosome, the gap junctions, and tight junctions, all shown on the same cell. Let's talk about a gland. Now, when we think of a gland, I want you to think of something that makes a secretion. And there are different ways to make secretions. So right now, glands make secretions. Later on in this presentation, we'll talk about the different ways we can make secretions. Some of these secretions stay in our bodies. and then some leave our bodies or are eliminated from the body. So when we use this term eliminated, this means to leave our bodies. Now, we're going to do some differentiation right now with terminology. These secretions, when we make a secretion, it's typically going to be something that's considered useful for our body. An excretion is something that we want to get rid of. It's a waste product. I don't know if any of you grew up in an agricultural or a rural background. I grew up on a farm. The difference between soil and dirt wasn't necessarily the composition, but where it was located. Soil was located in the field. Dirt was on the kitchen floor. And it's kind of like secretion and excretion. They can be the same chemical compound. When that chemical compound is serving a useful purpose, we call it a secretion. When that chemical compound is something we want to get rid of, we recategorize it as an excretion. Now, there are two broad categories of glands in our bodies. We have endo and exocrine glands. When we think of an endocrine gland, I really want you to think endo for in. Endocrine glands secrete chemicals that stay in the body. How do these chemicals stay into the body? Well, they're secreted into the bloodstream. And these chemicals that the endocrine glands secrete into the bloodstream are going to be categorized as hormones. When we think of a hormone, there are two broad kinds of hormones. We have steroid-based hormones. That's a kind of a lipid-based hormone. Or we have protein-based hormones. And these hormones are going to be used as chemical messengers in the body. You can think of them as a way to send information from one organ to another organ using the bloodstream as a means of communication. For those of you who still handwrite letters, you can think of an endocrine gland as the slow communication. It's kind of like sending a letter in the mail or snail mail. So it's typically a little bit slower of a process for communication, but it's still a very effective process for communication. We also are going to have exocrine glands in our bodies. And as we think of exocrine, I want you to think exo, exit. These exocrine glands are going to make a secretion that leaves the body. Now, for the exocrine glands that are on the surface, like our sweat glands or sudoriferous glands and our tear glands, also known as our lacrimal glands, these secretions leaving the body makes a lot of sense. But there are also internal exocrine glands. I'm going to say that again. We have glands inside of our body that make secretions that leave our body. And usually for a lot of students, this is kind of a sticking point. So I need to camp on this concept for a little bit here. When we look at the pancreas, when we look at our salivary glands, when we look at glandular secretions of our digestive tract, those secretions that are put into our digestive tract eventually leave our body. It usually takes about 12 to 24 hours for those secretions to leave our body. But eventually, when you go to the bathroom, those secretions will leave our body. So the mucus of our digestive tract, the digestive enzymes of our digestive tract, all of those secretions that go into the GI tract are categorized as exocrine secretions. Now, as we look at the glands in our bodies, we also have kind of the odd duck out, and these are going to be the unicellular glands. These unicellular glands are found in epithelium, and recall epithelium is typically used to make membranes or protective barriers, and they're non-secretory glands, generally speaking. So when we think of like a goblet cell within the pseudostratified columnar epithelium of the trachea, that goblet cell is a gland that makes mucus, but it's a one cell, it's a unicellular gland. If we look at our stomach, we have parietal cells and chief cells and the gastric pits of our stomach. And in addition to making secretions that go into the stomach, these cells can also make secretions that go into the bloodstream. So they can have both an endo or exocrine function. Let's move on to the next slide. When we look at a general or a typical exocrine gland, this is going to be a gland that makes a secretion that leaves the body. So to reinforce, exocrine glands make secretions that leave the body. These exocrine glands are going to have a capsule around the outside. This capsule around the outside of the gland helps to keep the secretions from going into the body. This capsule is going to allow for the gland to be segmented. This also is going to allow for the fluid within the gland or the secretion of the gland to be funneled to a common opening. We're also going to have stroma. And when we think of the stroma of an exocrine gland, I want you to think of just the structural framework. The stroma of our exocrine glands are going to support the secreting cells of the gland. So the capsule is the membrane around the outside. Stroma are the tissues of the gland that give it structure and shape. And then we have the parenchyma. The parenchyma are the cells that actually make the secretion. So to reinforce this idea here, we have the capsule around the outside, the stroma gives support, and the parenchyma makes the secretions. So these are generic terms that can apply to any multicellular exocrine gland. As we look at these glands, we can categorize these glands by their shapes, both by the shape of the duct and by the shape of the gland itself. So here we can have a duct that's just one straight opening. So that's a simple duct because it is an unbranched duct. Over here we have 1, 2, 3 ducts merging to one opening. That would be a compound or branched duct. And then we can also categorize our exocrine glands based on their shapes. If they are tubular, the secretory portion, the parenchyma of the gland, are going to be just a straight, narrow tube. If they are a cinar, I want you to think of little cul-de-sacs, for lack of a better term. These acinar portions are going to be little dead-end sacs that make secretions. And then we can have kind of the in-betweeny category, the tubular acinar, where we have dead ends that also kind of merge together, and the length of the tube itself also is going to make secretions. So here we have a simple tubular. We have one straight shaft to get, or one straight duct and one straight secretory portion, a simple tubular. Here we have a compound acinar. So the tube, or the duct, has branches, so we call it compound, and then acinar, the secretory portion, are the blind dead ends, or the cul-de-sacs. And then we can have a compound tubuloacinar. And in this situation, we have multiple branches for the ducts, but the duct itself also is going to be making secretions in addition to the dead end sacs, or the cul-de-sac. So this is going to be our most complicated kind, this tubuloacinar. We can find tubuloacinar in the pancreas. We find lots of compound acinar in the mammary glands. And then the simple coiled tubular is by far the most common. When we think of our sudoriferous, or sweat glands, they are very frequent within the epithelium of our bodies. We also have serous glands. When we think serous, I don't want you to think serous membrane. In this context, I want you to think watery or liquidy, very liquidy, highly runny secretions. Serous glands make runny secretions. So when we think of sweat, milk, tears, and our digestive juices, those have a high water content. Those are going to be serous secretions. Mucus glands are going to make a glycoprotein known as mucin. And this glycoprotein, called mucin, when it mixes with water, becomes mucus. You can buy powdered mucin, throw it into a bathtub, and have a bathtub filled with snot. It's kind of cool, actually. These mucus glands, the most common mucus gland is going to be a goblet cell. And as I mentioned on a couple slides ago, a goblet cell is an example of a unicellular secretory gland. And then we can have our mixed glands. A lot of glands in our bodies are going to be mixed glands, in that they are going to make some watery secretions. They'll make mucus or thick secretions, and the watery secretion will mix with the mucus or thick secretion. So let's move on to how we can make secretions, modes of secretions in our bodies. We have apocrine, merocrine, and holocrine. We'll start with apocrine. When we look at apocrine, I want you to write in the margin notes right now, budding. When we have an apocrine secretion, we have a budding of the chemical. So the chemical we're trying to secrete builds up on the inside of the cell membrane, the intracellular side of the cell membrane, and then the cell membrane itself bulges out, pinches off, and then we have a large transport vesicle that's extracellular. So we have a lot of phospholipids that make up the membrane, and then we have the chemical inside of the membrane, inside of this extracellular transport vesicle. So when we think of an apocrine secretion, I want you to think of a secretion that has a lot of lipids and that has a lot of proteins. So this is a protein-rich and a lipid-rich secretion. So now that we've talked about the modes of secretion, this protein and lipid-rich secretion, let's connect this to something that we all know about, milk. When we think of milk fat from the mammary glands, this comes from an apocrine secretion. We have cells in the mammary glands that are going to take proteins, in this case a casein protein, bind it up, and then cause it to be butted off and wrapped up in lipids and then secreted extracellularly. We use this term apocrine to describe axillary sweat glands, or sometimes referred to as urogenital sweat glands as well. These are I always call these the stinky sweat glands. We used to think that those glands in our armpits and down by the genitals and the perineum area made their secretion via apoptosis, but now we know that those glands actually go through a different process known as merocrine, which we'll talk about in just a moment. Like right now. So when we think of a merocrine secretion, these are going to be used by the ecrine glands of our bodies. And when we think of this merocrine secretion, I want you to think of exocytosis. So I want you to take this term merocrine and connect it to exocytosis. Let's go back to apocrine. If there was a single term or word I want you to associate with apocrine, it is budding of the secretion. With merocrine, we have exocytosis of the secretion. And then finally, we are going to have our holocrine secretions. When we think of holocrine secretions, I want you to think of cells going through apoptosis. The cells that are going to be producing the secretions will actually self-destruct and disintegrate. So if these cells are disintegrating, that means all of the macromolecules that are present in the cell are going to become part of the secretion. So we're going to have all of the lipids, all of the carbohydrates, all of the proteins. Holocrine secretions are going to be secretions that are very rich in macromolecules and very nutrient-dense secretions. So when we look at our skin, our oil or sebaceous glands in our skin are going to have holocrine secretions. When we think of our eyelids, we have a lot of, in our eyelids, proteins that are antimicrobial. Those antimicrobial proteins are oftentimes going to be secreted via holocrine secretions. Let's move on and focus on membranes. When we think of our membranes usually are going to be epithelial in nature. Sometimes they can be connective, and sometimes it's just a mix of different tissue types. When we think of connective tissue membranes in our bodies, we have the dura mater in the brain, we have synovial membranes in our joints, and we have the periosteum wrapping around the outside of our bones. We also are going to have epithelial-only brains. Think, for instance, the cornea of the eye or the lens of the eye. Those are epithelial-only membranes. And then we can have the skin, which is kind of a membrane and an organ all at the same time. It's an organ because it has multiple tissue types working together for the same purpose. It's also a membrane because it serves as a barrier in our bodies. Our skin has multiple layers to it. We have stratified squamous epithelium in the epidermis, and then that connective tissue underneath is going to be dense, irregular connective tissues making up the dermis of our skin. We also can have mucous membranes or mucosas in our bodies. So right here, this is a slice through the human trachea. And as we look at this human trachea slice, we can see that there's a pseudostratified columnar epithelium. Then we're going to have goblet cells producing mucus, or mucin, that mixes with water and becomes mucus. And then we have cilia that move the mucus around and up and out. Directly beneath this epithelium, there's epithelial tissue that needs nutrients. We're going to have the submucosal membrane. And this submucosal membrane, or lamina propria, is going to have lots and lots of blood vessels in it to supply lots of nutrients that will diffuse through the basement membrane to these pseudostratified columnar epithelial cells. One of the things I appreciate about this slide is that when we look at this figure from your textbook, you can see the part of the goblet cell right here where my little laser pointer is that we can often see when we look at our microscope slides. But then you can see the skinny, tiny shaft that includes the nucleus of the goblet cell. So in this figure, it actually looks like a wine glass or a goblet cell. Even though when you look at it in a lab situation on your microscopes, you really can't see the tiny, skinny shaft. All we see is that large region right there filled with the mucin. We also are going to have serous membranes, or the serosa of the body. We talked about those with our Unit 1 exam and our Unit 1 material, particularly when we were covering Chapter 1. So when we think of a serous membrane, let's review again briefly. There are two layers to a serous membrane, the inner layer and the outer layer. The inner layer is called the visceral serosa. That outer layer is called the parietal serosa. And as we look at the serosa, it's going to be a simple squamous epithelium, and the simple squamous epithelium is going to be secreting liquids. These liquids are very watery and slippery. It's a serous secretion. As we look at these serous membranes, we find that they line most of the major organs of the body and surround and cover them. Inside of our blood vessels and heart, we have a very smooth, slippery serous membrane. When I would do heart dissections with my students, I would make it a point to have my students take their pinky and shove it into the left ventricle and then shove it out of the aorta, and they can feel how slippery that endothelium is that lines both the blood vessels and the heart of our body. And then when we look at the mesothelium, that mesothelium is going to be our traditional serous membrane. So the pericardium, the peritoneal, the pleural, the parietal pleura, the visceral pleura, these are those traditional serous membranes that we'll talk about in Bio 314. But all serous membranes are going to have the same generic structure. We're going to have a simple squamous layer of cells with some supportive tissue underneath, in this case areolar tissue on our figure. And then that areolar tissue will be highly vascularized. There'll be a lot of blood vessels in that areolar tissue layer supplying nutrients to the simple squamous epithelium so that that simple squamous epithelium can secrete the watery serous fluid. And that's my note to stop recording. So we are done.